Thank you, everyone. So as Scott mentioned, I am sharing this talk with Paulina, and I would like to say that the previous speakers, I think, set the ground very nicely for us, so we don't need to convince you guys much the importance of what we are going to say. So just let me think that the complexity of these two samples poses a daunting challenge for the analytical techniques and also for our desire to control the stability and homogeneity of those materials. For us, as also Christina pointed out very nicely, the definition of some of the parameters that are important to us, homogeneity means that the microbial taxa and metabolites are the same across different values, right? The stability means that the microbial taxa do not change, and also the metabolites do not change over time. So the ideal preservation method will leave us with intact cells, and the integrity of the cell wall will not change. So based on that, so what are the factors that we should consider in order to make a reference material that is stable? That it's stable and homogeneous. And this is what our talk is about. So our pilot study, in our pilot study, we were interested in understanding a little bit the impact of the storage condition on the profile of the metabolites and also the homogeneity across different values. So as a pilot study, this is, we have just preliminary data, so we are just scratching the surface of this huge effort. And our samples, they were composed from, they came from a single donor, and the sample were split in 4 different vials and stored in these 4 different conditions. So lyophilized 4 degrees, water 4 degrees, water -80, and the 2 buffers, proprietary buffers that we call A and B. I wanted to point out that at this point, everything that I heard about is to sample in the gut microbiome. So usually the preservation method was always targeted the DNA. It doesn't matter what happened with the cells as far as the DNA are intact. So now I think it changed a little bit the picture. This material was stored. So once the Biocollective they prepared, so it was sitting in the respective conditions for 4 months. So once we were ready to analyze, so they were split in different aliquots, and the material were processed using NMR, GC, ILSI, and Metagenomic 16S. I am going to start with the NMR data, right? So this is the protocol that I use to extract the metabolites. I start with 100 milligrams and use 400 microliter of dilute water, and vortex spin down and transfer to NMR tube. The syringe, because the syringe that I use to filter, I use the same one, pass methanol to dissolve some of the compounds soluble in methanol, and I put in my pellet, extract vortex, spin down, and transfer to a tube for the analysis. So one thing that I noticed for the 2 buffers, I obtained these 2 very nice clean solutions. For the other samples, I had this floating solid on the surface after the centrifugation, which is an indication fat and maybe lipids. The analysis, I use CPMG pulse sequence to suppress the broad peaks that comes from micromolecules and lipids. I also measure the pH of the samples because this causes shifting in the NMR. Of course, that the pH is something that you can correct by adjusting the pH, but I just want to see how the whole thing would look like. So I didn't correct for any pH, which was interesting to see. So there is a difference in there. So going to the result, so I think the main thing that I noticed was the impact of the buffers, right? So the privatization buffer, they had the multiple components in high concentration that significantly hinder the metabolite analysis. So it's these first two over there, so they have ethanol and they have other things in there. In addition, so the buffer A had a pH of 9.74, and the other one, it was about 6.2. So this left me, in addition, so I also noticed that the samples with the buffer, they were the one that had the broad peaks there. So only these two samples. So as I said, this left us with the other three samples. And the 4 degrees, this one on top, so the pH, it was the lowest pH compared with the other samples. So this actually caused a little bit to shift in some of the peaks, but not all of them. But I wanted to point out that it was not really just a shifting peak, but some of the metabolites there, they were very different from the other two samples. If you look at minus 80 and the lyophilized one, look at two different regions, so they were very, very similar in terms of the metabolites profile. In the case of the 4 degrees, so it exhibits significant differences compared with the other two samples. Moving forward, we look also at the repeatability of the samples, right? So looking at vial, cross vials, so how they look like. So here it's an example of the lyophilized one. Also look at the two regions. They are very similar. If you put this on a PCA with all the samples, I just wanted to point out that for each condition of the three samples they analyzed, they were very, very close. So they are very apart, which means I think it's a little bit misleading because most of the difference that comes from the buffer A and B, in fact, is related with the components of the buffer and not with the metabolites itself. If you expand region A, you can see that the 4 degrees, the lyophilized and the minus 80, they're still very close, and the water one, it's a bit different. And I have one of the data that it was a little bit apart, so I'm trying to look into this as well. So some of the components, they are the usual suspect when you have extraction in water. So I forgot to mention that although I collect 1D and 2D spectra and also the samples in ethanol, unfortunately I didn't have time yet to analyze those samples. And I think that's it. I'm not going to do some of the considerations because Paulina will do that in the end as well to get a general idea. Thank you.
Thanks. So in addition to some of the NMR characterization, we also conducted more traditional mass spec type metabolite data. So the gas chromatography mass spectrometry system is kind of your typical volatiles analysis. And we performed this with solid phase microextraction, in which you take a fiber that is coated with a sorbent. In this case, we used a triphase sorbent to try to get a kind of the largest scope of compounds possible. We incubate our sample at 60 degrees for 40 minutes to allow the components in the sample to volatilize and sorb onto the fiber. We then introduce that fiber into the gas chromatography injection port, which is at a very high temperature, thus desorbing the compounds off the fiber and introducing them into our system. So a question was raised earlier in terms of how to preserve the volatiles, and we actually found really interesting results in terms of that. So this is an overlay of the 4 aliquots of the lyophilized material. You could see that overall they're relatively reproducible, they're overlaid, and we do retain those really volatile fatty acids. So we still see acetic acid, we still see propanoic acid, butanoic acid, and so on and so forth. We also see some of the other components that we would expect to see in stool samples, such as indole and 3-methyl-indole, which are common E. coli metabolites. When we compare across our 4 different preservation conditions, you can see that it's kind of a modge podge. Everything looks very different, indicating that the different preservation techniques result in different metabolic profiles. And one thing that kind of comes off really surprising to us is the signal in the lyophilized samples is still the highest amongst all of them. And I think what we think is happening in this case is the fact that the lyophilized material is kind of powdered, so there is greater surface area for the desorption of the compounds to happen, as opposed to the liquid samples, which have a more limited surface area scope. Another thing that we've sort of concluded from these data is the fact that the 2 proprietary buffers, we believe that they are lysing the cells, because those metabolic profiles look way more complex than those that are stored in water. And I think that one of the questions we really would like an answer to is, are we interested in solely the intracellular metabolites, or are we also interested in the extracellular components? Or I think I got that backwards. So are we interested in solely the extracellular metabolites as opposed to also the intracellular ones? Another thing that we see is the fact that the lyophilized samples are grouping kind of far away, and once again, I think that that is a surface area analytical difference as opposed to a sample difference. But the water and the 2 samples stored in water at 4 degrees and at -80, in terms of metabolites, look fairly similar, and that's probably due to the relative ratios of compounds being similar in this analytical technique. We also performed liquid chromatography mass spec, which is kind of your gold standard metabolomic analysis. For this analysis, we performed extraction with water, methanol, and acetonitrile. So we added the solvent. We would vortex the sample, centrifuge, then subsequently add the next solvent, and we combine all 3 extracts. And the data analysis, so the analysis was performed on a C18 column, which is kind of a very generic analytical procedure. And we processed our data with MS-DIAL and did spectral library matching in the NIST MS library. So keeping in mind that this is very preliminary data, we don't have a NIST level confidence in the identification of these components. So once again, looking at an overlay of the different storage conditions, once again, there is kind of no coherence. They all look very different, indicating that storage plays a very important role on what metabolites we can detect. So in the liquid chromatography analysis, we see some of the larger biomolecules, things that have, like, fatty acids with a more of a cholesterol backbone, and also some of the kind of longer chain saturated fatty acids also. One of the proprietary storage buffers was absolutely not compatible with ILSI mass spec analysis. So the DNA stabilizing agents that were present basically overwhelmed all of the signal we were able to obtain. It led to a contamination of our instrument, and so we would really urge not to store your microbiome samples in a proprietary buffer if you also plan on performing metabolomic analysis. Lastly, we also did some of the metagenomic analysis with 16S RNA. So the DNA was extracted. I believe it was just done according to a commercially available kit. So the PCR was performed in triplicate and then ran on a MiSeq to do the sequencing, and then data was analyzed with data2, and once again, as with all of the data that we have presented, this is very preliminary. One of the things that was notably observed was the fact that the samples that were stored in water in 4 degrees were very much different from the other storage conditions. The third technical replicate in one of the proprietary buffers was very different also, but we believe that that is an instrumental problem as opposed to a difference within the samples themselves. And once again, this kind of concludes that we really do need to pick a preservation technique wisely. What we think is occurring is that the samples that are stored in water and at 4 degrees have continual bacterial growth during storage, therefore changing the taxonomic profiles that we see. So to conclude our talk, one of the things that we've kind of came across is that within a preservation technique, we have really good reproducibility amongst all of our analytical techniques. We believe that they are sufficiently homogeneous and that the Biocollective does a really good job homogenizing their samples. We did observe differences among storage conditions, and some of those differences were different based on what the analytical techniques that were used to analyze them. The proprietary buffers obviously had components that interfered with the NMR and mass spectral measurements. Buffer B was kind of detrimental to our LC-MS system. And one of the things that we've observed is that the buffers may lyse the cells, and that's a question that we want the global community to answer, is whether or not that is important or not. And while this wasn't a true stability study, we have evidence to suggest that bacterial growth may be continuing during storage, so that is obviously something we don't want for a homogeneous and stable material. So with that in mind, some of the final considerations that we've kind of come up with. So amongst the different preservation techniques, one of the things we saw is pretty dramatic differences in pH, likely due to the fact that water at 4 degrees was more acidic, likely due to CO2 infiltration. One of the proprietary buffers was on the other side of the spectrum, was very basic, and that's probably due to the components within the buffer. For metagenomic analysis, we had pretty high suitability amongst the different preservation techniques, with the exception of water in 4 degrees. We don't want to see bacterial growth occurring. In the metabolomic space, we had low suitability with the proprietary buffers, and obviously we had the potential for bacterial growth, potentially some in the lyophilized samples, also because they were stored at 4 degrees. Water, we saw high potential for bacterial growth, and less so with the -80 storage and the proprietary buffer storage. And once again, the potential for cell lysing, lyophilization, and storage at -80 can be pretty harsh on cells, as opposed to water at 4 degrees, not so much. And then obviously we are most likely lysing the cells in the proprietary storage solutions. So with that, I'd like to thank everyone else at NIST who is involved in this project. This work would not be possible without them, and the Biocollective for providing us with the samples. With that, Sandra and I will entertain any questions.